This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 53 for October 5th, 2009. Hi, everybody. It's Vincent Racaniello, and I'm here in New York with Dick Despommier.
Hey, Vince.
And Alan is filtering through on the Skype line from Western Massachusetts.
Buongiorno.
Buongiorno, come stai?
Bien, you said.
Silence.
Yeah, you reached the end of my Italian.
That's the end of your Italian. How are you, Alan?
I'm doing okay.
And Dick is just back from India.
That's true.
I thought you were going to be back tomorrow.
So did I.
So we're actually recording on a Monday, which is unusual.
It is.
Because I'm going to be away the latter part of the week.
That's right. Giving out government money to the...
I'm going to be reviewing grants for the government, yes. Alan, you're coming to New York tomorrow.
Yes, yeah, I'm going to be covering a conference about lupus clinical trial design.
Nice.
Tough field to be in.
I'll say.
And, so they're having a post-mortem on some of the trials that failed and the one that worked recently. So they're
the one that...
Sounds like AIDS.
It's an autoimmune disease that's been difficult to treat. There's a bit of a running theme there, isn't there?
Yep. Well, I just spoke with Scott Hammer last week, and that's TWiV 52, about the AIDS vaccines and trials, because he's involved in a lot of those.
And tribulation.
Check those out. And a couple of things before we get into our science. A few of our listeners have started making transcripts of TWiV. We just have a few posted, but they're up there. Take a look.
Wow.
Anybody that wants to help. It's not easy work. It's not for everyone, but it's appreciated. By the way, I heard that Dick, you listened to TWiV on your iPod on the long flight.
I did.
And I heard you like it.
I was repressing my laughter.
Well, you should go to iTunes and write a review.
Maybe I should. That would sort of be self-serving, though, wouldn't it?
I thought that was amusing that you liked it. Have you ever listened to TWiV, Alan?
Yes, I have. After when I first joined the show, I listened to a couple of episodes just to make sure that my sound was good. And but to tell you the truth, I don't generally go back and listen afterwards because, I just... something about listening to stuff that I already did.
Yeah, I feel the same way, but I actually, with this long plane ride, 17 hours, and you run out of things to do, and it just happened to have been copied onto my iPod tunes.
By mistake, right?
By mistake. I'm sure it was. I said, wow, I've got 2 episodes here, and I clicked them, and it was a lot of fun just to listen to the old broadcasts.
Well, I listened to them for several reasons. I think reinforcement, it never hurts. Mm-hmm. I also want to make sure we got everything right.
Sure.
And I want to make sure the sound is good, because there's nothing like it. But, when I edit it, I end up listening to the whole thing anyway.
Exactly.
So I do a few times, but I don't mind.
Well, we appreciate the hard work that you put into this. I like
the sound. It sounds good.
Well, maybe on the train ride to and from New York tomorrow.
Just pick one, and maybe one that you weren't in, and you can listen to it.
Right.
And we should say that TWiP, is that it? TWiP this week in parasitology.
Yes.
Is gestating.
It is.
Should be around in a couple of weeks. It's incubating.
That's right. This is the pre-patent period. We'll get to that.
Dick and I are doing a parasitology.
Yeah.
It's going to be monthly, but we decided to call it TWiP anyway.
Sure.
Because TMIP doesn't really.
Exactly.
No.
Yeah, we went through that. TMIP just didn't cut it.
That's right.
So that's fine. Okay, and now, today we're announced the Nobel Prizes in Medicine.
Yes.
Elizabeth Blackburn, UCSF, Carol Greider from Hopkins, and Jack Shostak from Harvard.
Congratulations to all of
Telomeres and Telomeres.
Wow.
That's pretty cool.
Very.
And now that they've won the prize, we know that the ends justify the means.
Excellent. Did you just come up with that spontaneously?
Yes, I did, as I was just looking at the notes.
That's
justify the means.
Fabulous. That is
Dick,
fabulous.
Do you know what this is all about?
I do, actually. I have a segue here that leads into a book that I helped produce called Younger, Younger, written by Judith Sulzberger, who is a graduate of our medical school. And it was based on the scientific principles that these 3 people received their Nobel Prizes for. Namely, that as the telomeres get shorter.
What's a telomere, Dick?
Telomere is the end of the chromosome.
Okay.
And it's a series of repeated units. And each chromosome has an X number of repeats. And telomerase, of course, it's almost like a death sentence for the cell. As soon as the last repeat is clipped off, the cell can no longer replicate.
We should make clear that you lose your ends as the DNAs replicate.
You do, as the
we
cells
should also
divide.
make clear that that was supposed to be the sound of a ticking clock.
Well,
those were not hoofbeats.
No, we're not. And I apparently am under the impression, at least, that some cells of some tissues have longer telomeres than other cells in other tissues, but that's...
And also, for the sake of some of the audience who might not have taken this in a molecular biology class, if you look at the way DNA replicates, if you have a linear molecule of DNA, you have a problem when you get to the end,
right?
Because you have a template for the last little bit of one of the strands, and so you lose a little bit of the end.
Yep.
And if you do that through generation after generation, you're going to lose a little bit, and eventually, you're talking about real genes that you're cutting pieces out of.
Exactly.
So the telomeres are just this spacer sequence that's stuck onto the end, and the telomerase is the enzyme that sticks that spacer sequence onto the end of the chromosome as insulation.
So this book takes that principle and then says, what happens if you could make them grow back?
Science fiction.
Could you reverse the aging clock?
That is exactly right. So the book itself is called Younger because it depicts the life of a virologist and a neurologist. And they form this research consortium to investigate how to transfect a virus that carries the gene that reverses the telomerase process back to a longer telomere. And they've tried it in mice first, and of course, I don't want to spoil the story for you. It's available on Amazon.com. It's a very well-written book, and in fact, it won a bronze medal for independent publishers for the best science fiction book of, I believe it was 2004.
Now, I understand you can skip the last 5 chapters because they're all the same.
But if you do, you'll die at the end.
Oh, right, yes.
So that's your pick of the week, right?
Of course it is.
Wow, so we did that early on today.
We did.
It's okay. So telomeres are those funny ends, and the telomerase is the enzyme that puts them back, and these are the folks that discovered them. And we get old in part because we lose the ends, right?
Apparently so.
And what happens, Dick, if you reactivate telomerase in a cell?
Well, tell me what happens.
It gets transformed. It can become a cancer cell, right? So that has some application here. Anyway, Dr. Grider worked with Dr. Blackburn, which is an interesting sequence here, and Dr. Shostak is independent.
We had something like that happen here at Columbia, too.
Who's that?
It was the Nobel Prizes that were given out for looking at the molecular biology of smell.
Yes, Richard Axel.
And his graduate student.
Can you remember her name?
I can't.
Linda Buck?
I've never met her. Linda Buck is exactly the name I was
Alan.
Seeking in my memory bank, which has disappeared.
She was a record-setting graduate student.
Yes,
really?
Over 10 years.
No kidding.
No kidding.
Wow.
People like that were an inspiration to me when I was...
Well, you didn't take that long.
No, but I always reminded myself that, well, at least I haven't done that. But then again, I didn't win the Nobel Prize.
No,
either.
you didn't.
Maybe I should have spent 10 years in graduate school.
No, you don't second-guess yourself. So anyway, we thought we'd mention those, even though they're not virologists.
No.
But good science nonetheless.
Very good science.
Which is what TWiV is all about.
Exactly.
Pardon me.
Apparently, we're both congested for different reasons.
I had a small upper respiratory tract infection, which I'm attempting to culture in the laboratory as we speak.
Really?
Yeah, see what it is. All right, let's go on. Our first story was something that Alan Dove discovered several weeks ago. Bacteriophages encode factors required for protection in a symbiotic mutualism.
Yes.
Right.
Now, it's important to note I discovered the paper, not the...
Right.
So I did the easy part.
Right.
Would you like to discuss this?
Sure, I'll introduce it at least.
Okay.
Be interested to hear your thoughts on it as well. So this came out at the August 21st issue of Science magazine. As Vince said, I put this up there a few weeks ago, but we hadn't gotten to it. But this group, Nancy Moran and colleagues, they're at a few different places, University of Georgia, University of Arizona, and they were looking at one of these obscure basic research sorts of systems that people don't pay a whole lot of attention to. And they found something really, really cool. So they were looking at aphids. And these aphids, the particular aphids they're studying, are attacked by a parasitoid wasp. This is a fairly common thing that happens in nature, especially among insects. You get a wasp that parasitizes a particular insect, lays its eggs, the larvae hatch out and kill the larvae of the wasp hatch out and kill the larvae of the host. And I think we've talked about this before. There are some interesting biological control possibilities in there, and these folks discovered that there's a symbiont, a bacterial symbiont inside the aphid. And in bacterial symbionts, we know that there is often a bacteriophage, a virus, that's responsible for controlling, some of the pathogenic aspects of bacteria. But in this case, we've got a bacteriophage that's controlling the symbiotic characteristics. So you've got a symbiont. I kind of wove through this story in an odd way. So you have an aphid. It has a bacterium living inside it that is symbiotic. That's protected. Turns out that bacterium protects the aphid from attack by this parasitoid wasp. The key to that protection is a bacteriophage infecting the bacterium. So you've got a phage inside a bacterium inside an aphid that protects the aphid from being parasitized by a wasp. Did that make sense?
Got it.
Yes.
Yeah.
Yes.
Okay.
Yeah.
So you have to raise the question, why would anyone be interested in aphids, or aphids, as you pronounce them?
Which is the right way? Aphids?
Aphids is probably correct.
I always called them aphids.
To learn things like this.
No.
Are they crop
They're
destructors?
plant pests.
They're
pests.
serious plant pests.
Also, do they prey on bees also? Or those are mites?
Those are mites.
Are they
are mites.
related?
No.
Not at all?
No, mites are arachnids, in fact, and these are insects.
So what do aphids do to plants?
Yeah, they eat
They
them.
suck juice out of them.
Suck the
That's
juice
right.
out?
That's right.
So the wasps are doing a good thing by killing the aphids.
It depends on the aphid, of course.
Right. There are some aphids that you probably want to have around.
That's right.
What I really want to know is, how does the wasp see the bloody aphid that's so small?
Exactly.
Does it smell it?
In most,
Probably.
in the cases that have been studied, parasitoid wasps will often have some receptor for a pheromone that's used by the aphid.
Well, this aphid is not much smaller than the wasp.
That's
actually.
right.
There's a nice
That's
picture
right.
here. This wasp curving its abdomen around to
Yeah.
inject this thing.
It's
This is amazing. And the aphid
you're
is just
you're
pink.
absolutely right about this. I mean, the beauty of nature at that level is absolutely stunning.
So the aphid gets the wasp larvae or egg injected into it, and most of the time the egg hatches and it kills the aphid.
And right, the larvae. This was the inspiration for the movie Alien. The larvae eat their way out.
Probably.
Yeah, I think explicitly. That was,
Sure.
Ridley Scott even said that at some point.
Interesting.
That the larvae eat their way out from the inside.
Yeah.
But there's tons of examples. I mean, these wasps that catch spiders and just stun them and then put them in their hole and lay an egg, and the next thing you know, you've got another wasp. So,
Yeah.
I mean, we've known about these relationships for quite a while, but
Yeah, this is a very common wasp lifestyle. And people
Yeah.
think of wasps, they think of the yellow jackets and hornets.
That's right.
But they're actually, the great diversity of wasps are
Yes.
going around parasitizing other organisms.
sure.
So they in this paper they took aphids with bacteria lacking this bacteriophage and they did
Right.
parasitism bioassays. I presume they
Cool.
let the wasps lay their eggs in them and see the outcome. It turns out that this bacteriophage encodes a toxin, which kills the wasp larvae, I suppose.
Yeah.
And remember, bacteriophages are viruses of bacteria.
Right.
Many of them integrate their DNA into the DNA of the bacteria. In fact, we knew about integration long before we knew about retroviruses, right?
Yep.
Yes.
So actually, that's probably not true. We knew about retroviruses in the early 1900s, but we didn't know they integrated.
We didn't know what they did.
And we knew
Right.
that bacteriophages integrated into their hosts long before we knew about retrovirus integration. So that's what this bacteriophage does. So I presume the bacteria benefits from being inside the aphid, and when they have this bacteriophage in them, it benefits the aphid. But
Right.
my question would be, what about the aphids without the phages, just the bacteria? What's the benefit for the aphid there? Maybe none.
Maybe.
Well,
because they
Maybe.
get killed by the wasp.
In some cases, there are some aphid species that have symbionts for digestion.
Right.
Yes.
So I don't know if this bacterium is one of those.
Some bacteria also determine the sex of the insect.
Mm-hmm.
They actually turn the sex of the insect around. So Wolbachia does that. I think we've brought this up before.
Yes, they talk about that here. I have a question. So you would think eventually you would select for all aphids to have bacteria with the phages in them. Otherwise, they would be eliminated. Unless there's a negative phenotype or a negative selection for having the phage as well, which there could be.
Right.
I mean, there's an energy consideration here too. How much energy does the aphid spend keeping the bacteria alive?
Mm-hmm.
And is there any feedback between the bacterial metabolism and the biological needs of the aphid? You say bananas and I say potatoes.
We have an email about bananas, by the way.
Oh, good.
So this is a very nice paragraph here. There are examples of bacteriophage encoding virulence factors for pathogens, such as Shiga toxin in E. coli, cholera toxin in Vibrio.
Yes.
And this is the first instance of phage encoding factors underlying a mutualistic symbiosis. That means both ways, right?
Right.
That's correct.
Very cool.
They forgot to mention diphtheria, too. That would have included that into the pathogenesis factors for bacteria.
Yeah.
Cool stuff.
The phage is implicated in conferring protection to the aphid and thus contributes to the spread and maintenance of the bacteria, H. defensa, in natural apissum populations. Apissum being the aphid. Right? Is that correct?
Right.
In general, phages offer considerable potential as vectors of functional change within and among bacteria.
Right.
It's very interesting. Now, this is beautiful work, but it's becoming increasingly difficult to support.
Tell me about it.
Because as the money gets,
Well,
you know,
you'll find that out when you get to
less and
Washington
less.
later this week.
well, this sort of thing is not supported by NIH, which is National Institutes of Health.
This would be an agriculture department.
Right.
Which has even less money than the NIH.
Yes.
This is true. This is
It's a shame
right.
because this is made possible by incredible technology that we now have, and you can't do it. And there's so many neat things like this that you can do. But when money is limiting, you have to make choices. You know, we can moan about it all we want, but...
And cutting back to what we were talking about with the Nobel Prize, the reason that those researchers were looking at the ends of chromosomes, I mean, do you know why they were looking at that?
You'll tell us.
Because they looked at the way DNA replicates and they said, gee, there ought to be a problem replicating the ends. How does that work?
Right.
Which at the time was an obscure problem, right?
It was an obscure problem that, who really cares? It's not specifically related to a disease as far as we know, but they were just kicking around looking at basic research topics, and they found this telomerase and the telomeres and this mechanism going on. And at the time they discovered it, it was kind of like this paper we're talking about now. It was a very interesting and elegant piece of work and very satisfying in its own way. But you couldn't point to it and say, well, this is worthy of a grant because it'll cure disease X.
Sure.
And now we talk about telomeres in cancer. We talk about telomeres in aging. There was a whole discussion about why when Dolly the sheep was cloned, her telomeres were actually shorter than they should have been for age because they were the telomeres inherited from an adult cell.
Here.
So, they discovered essentially an aging clock.
Yep.
And the initial discovery of it was this sort of basic question-driven research.
Well, there's no question. You have to follow what's interesting to you, and that's how we get the best discoveries. The real problem is how to fund all this. It gets more expensive, and as Jack Shostak said, it's getting harder and harder to do science nowadays. You have more tools, but the money is making it's very competitive.
And what he's going to find, as all other Nobel Prize winners have in the past, they are not given any special privileges when they reapply for their own grants.
Well, the grant has to stand on its own, right?
Sure.
Yep.
No solution as of now. Now we move on to something a little more close to home, perhaps. Not Nobel Prizes, but a 14-year-old girl who dies after getting vaccinated with the HPV vaccine. So, Dick, have you heard this story?
Yeah.
Young lady in the UK.
Yep.
She got her Cervarix, which is the HPV vaccine, one of the names of the HPV vaccine.
Right.
And shortly thereafter went into the hospital and died. So, Dick, did the vaccine cause her death?
Hard to say, just based on what you told me. What else happened to her that caused some problems?
Well, many things could have happened, and that's an association. That's an association in time, right? The immunization with the disease.
Yeah, it was an unfortunate association.
But many people believe that if that happens, then the vaccine must have caused the death, and that's the problem with the anti-vaccine segment.
Problem with that argument is that how many other people at the exact same moment also got that vaccine that didn't die?
Yes, they were fine.
So you can say,
and in fact, this vaccination was part of a major campaign in the UK with Cervarix, and they, the other girls at her school got the shot, and some of them reported some symptoms, but nobody else died.
What was the cause of death?
Well, it says
cause of death turns out to be a tumor, which is, of course, in no way related to the vaccine.
And it was a tumor that was unknown, I presume, until her death. It
was, I believe it was. The follow-up on this has been a little bit sketchy, but it looks like it was a tumor in her chest, somehow caused heart failure. And as I say, I'm not looking at the autopsy report, obviously, but
so does that fact supersede the suspicion that this injection process led to her death? I mean, that should clearly show.
No, of course.
So this makes news because?
Well, it made news initially because she was vaccinated that day and she then promptly died.
Right.
It made news because the newspaper needs to sell newspapers, and they are jumping on this. Aha, look, this vaccine may have caused her death.
Well, and also it was an event of legitimate concern to public health authorities. They immediately quarantined that batch of vaccine. They said, well, okay, yes, this is a coincidence, it may be causative, it may not be, but in any case, we don't want to take any chances. So they set aside that batch of vaccine.
Sure.
And then went and looked at why exactly did this girl die, and then, as it turns out, she died of completely unrelated causes. And this probably would have happened that day or soon after anyway. And so I think it's legitimate to report on it. I would have liked to have seen a little bit more profile on the follow-ups. As it was reported the day of, and then there was one follow-up saying, "Oh, it was unrelated," and then just no additional detail. Something like this, you'd kind of like the follow-up to be a little bit more prominent.
Yeah, I think that's a good view of it. It should be reported, obviously. I don't mean to say it doesn't, but as Alan said, the follow-up has to include the fact that she had an issue. Here in this article that I'm looking at, which is from the BBC, it just said she had a serious underlying medical condition.
So how long do we have to wait for this vaccine to prove out that it's actually preventing the spread or the cause of cervical cancer?
Well, I think it's already having an effect.
I mean, is that a 10-year down the road period or a 5-year or a 15-year?
I think in 10 years you see, if everyone receives it who should, you have very little cervical cancer.
And in the other group that didn't receive it, of course, the rates are predictable.
Same, sure.
Right.
I think there's no question that this is going to have a major impact.
How many lives have been saved by this injection procedure? And you can actually calculate that.
Of course.
And then
it's always the balance of that, right?
Exactly right.
And there are side effects with this vaccine. Here I'm looking at side effects out of 1.4 million vaccinations, 4,657 adverse reactions, including headache, nausea, dizziness, sore arm, and rash.
Yeah.
And I'm willing to bet that all of them except the sore arm probably are just
from something else.
Yeah, they would have happened anyway,
Correct.
even if you hadn't. And that's why she would have died anyway, most likely,
right,
if she hadn't been immunized. And I just object to fueling anti-vaccine sentiments because that's dangerous.
Yes.
And we need to immunize against infectious diseases. There's no doubt about it. Right. And that's why I bring this up, because it has really nothing to do with virology, but it's just I don't want to trash the vaccines anymore. And this is immediately, what people would have done.
Right.
Before we go on, it's time for a word from the sponsors of this episode, our good friends at Citrix. If you have a long commute to the office, which I do, you could be working from home just one day a week.
Tempting. But not on TWiV Friday.
Well, if you got Skype at home, you could.
We'll work on that.
Yeah. and reduce your commute time and expenses by 20%. 1 day out of 5, that's 20%. All you need is reliable access to your work computer. You probably need some other things too, but that would help. So use the easy and highly secure GoToMyPC to work on your office computer. It's very simple, Dick. Here's what you do. You go to their website, you click a button, and it loads a little program on your computer. You get a login and a password. Then wherever you are in the world that has internet access, you go to the website, you put in your login, and boom, there's the desktop of your computer, assuming you've left it on, of course.
Naturally.
You can see all your files, all your folders. You can even run programs that are on your computer from wherever you are.
It's amazing.
Email, browsers. So you could have access to all that you need. So you could stay home once a week and avoid breaking down on the George Washington Bridge.
Yes. By the way, how is your car? We won't talk about that.
It's very expensive to fix.
I'll bet it was.
So we can have you try all of this for free for 30 days. You just go to gotomypc.com/podcast. Okay, go to mypc.com/podcast, and you'll have a free 30-day trial. We thank Citrix for their support of This Week in Virology. An interesting article, I think you pointed this out, Salicylates and Pandemic Influenza Mortality.
Yes.
Very interesting.
Yeah, so this is a hypothesis,
Right.
as so much about the 1918 flu, unfortunately, is. We don't have much in the way of data, but this is really interesting, I thought. So this is from Karen Starko from Burlingame, California. It's a viewpoint published in Clinical Infectious Diseases, and the hypothesis is that, okay, so the 1918 flu, there was this very high case fatality rate, and in fact, it's really the reason we're worried about the 1918 flu coming back is that the case fatality rate was so high among young adults, especially. And some of the pathology slides that we have from that time show that the patients had bacterial pneumonia, which obviously, you've got the flu, you get a secondary infection with bacterial pneumonia. It's 1918, you don't have antibiotics to treat this, and you die. Earlier in the pandemic, there were a lot of deaths that appear to have been viral. And you worry about those. How bad is this virus? And so what this author points out is that there was apparently a regimen, a treatment regimen that was promoted in 1918. The U.S. Surgeon General, the Navy, the JAMA, all recommended the use of aspirin, and specifically high doses of aspirin. And they anywhere from 8 to 31 grams per day.
Wow.
And it's
Die from a bleeding ulcer.
Exactly. So, it's 1918. They did things in a big way. So this recommendation came out, in the fall of 1918, just before a spike in the death rate from the flu in October. And this author looks at what we now know about salicylate intoxication, which is aspirin toxicity. And an overdose of aspirin produces a variety of problems, including bleeding, hyperventilation, pulmonary edema. So, and in fact, there was a recent study apparently that found pulmonary edema at autopsy in 46% of salicylate intoxicated adults. So they died of an aspirin overdose, and their lungs were all fluid filled. So the hypothesis is, well, maybe this high case fatality rate was iatrogenic. These people were potentially would have survived the disease, but then they got the treatment.
Wow. A lot of episodes like that, unfortunately. I mean, not just flu and aspirin. There's a lot of other evidence for overtreatment and death from the drug rather than the infection.
Interesting idea.
Yeah.
Of course, it's associative, just as the HPV-associated death was.
Yep.
Although we do know in this case that aspirin does have similar consequences. I don't think we'll ever know the answer.
Right.
Even if you had a mouse and you showed that you made influenza worse by giving it aspirin, it wouldn't prove that this happened in people. But
you'd need to travel back in time to get the data you need.
Just shows you how desperate they were for treatments also because of the lack of antivirals and things like that.
Yeah, these people had very high fevers, so they were giving them aspirin copiously.
Yep.
giving him, here, there's a quote here. His fever kept getting higher and higher. Aspirin was given to him by the half handful over and over. You don't even, one Tylenol every 6 hours, right?
Yeah.
Wow.
We're much more careful. So it's very interesting. And I don't know what will ever happen. That's it. But there are many of these theories of 1980. We know the virus is very virulent. The virus that we constructed, not we, but scientists constructed, is quite virulent, more so than many other strains. So that's probably part of it. All right, that's cool. Now, the last bit is I found an article in which a Canadian microbiologist says hand washing is not proven to prevent influenza transmission. So it's got a lot of press in Canada. And, he may be right that it hasn't been shown in a clinical trial to prevent transmission. But here's the thing. We know virus can last on your hands. We know you can get it from your nose to your hand, right? Very easily.
You can.
So why take a chance? It's so easy to wash your hands. And plus, it's good anyway to get the bacteria off your hands, right? So this is just confusing people. I don't know why a microbiologist would say this. It's not in anyone's interest to say something like this where you have such an easy thing to do. And for example, there's a blog called H5N1, and the writer, whose name is Croft, he writes, "Now I don't know what to think. Washing your hands presumably won't hurt, but what will actually do some good?" So I thought I would answer that. I'll tell you what will do some good. Get immunized, first of all, right?
Number one.
The other day I taught Dick's class in emerging infections.
Yes, you did.
50 students. I asked them, who's getting the swine flu vaccine? None of them raised their hands. Why? Because they think it's for high-risk people. So I told them they could get it free here, and maybe some of them will. But, the more people that get the vaccine, the less the virus is going to spread. It's very simple. The vaccine is coming out this week.
That's right.
Go get it.
Yep.
Now, what else can you do? You could wear an N95 mask. Sure, they've been shown in clinical trials to help. It's up to you.
Yeah, I'll mention something about that in just a moment.
But if you don't wash your hands, to quote Mark Crislip, you're being a dumbass.
Yes.
Right?
Or Semmelweis. That drove him crazy.
So I heard Mark Crislip on a podcast of his the other day, and he said he brought his son into the ER, and before they gave him an IV, they didn't wash their hands. And he said, you got to wash your hands. Semmelweis taught this to us many years ago.
That's true. And then no one listened to him.
No one listened.
And he went crazy.
Well, Mark says nowadays it takes 10 years for things to percolate through the medical literature, but
I think all the nurses know this.
We've known Semmelweis's teachings for 160 years, right?
And like you said, it couldn't do any harm, so you might as well do it.
Right. So I think you ought to wash your hands. You ought to practice good hygiene. Don't sneeze, out into the air. Right. Cover yourself up as much as you can.
It's called barrier medicine.
Wash your hands. Get immunized. It's the best way to do it.
That's true.
Don't worry about washing your hands.
And as for the N95 masks, I tried to post this. I put it on my Delicious stream, but I guess it didn't get through. There was just a paper in JAMA. They released it early, posted online October 1st, comparing surgical masks and N95 respirators to prevent influenza among healthcare workers.
Mm-hmm.
So it was a side-by-side trial at a hospital, I forget where the hospital was, Ontario, and they randomized nurses, 478 nurses, randomized into two groups. Half of them got surgical masks, just the regular tie-on surgical, sort of a handkerchief type mask, and the other half got issued N95 respirators and showed how to use them. So, they're going around, they're looking at regular seasonal flu outbreak, and they found that they got influenza infection in 23.6% in the surgical mask group and 22.9% in the N95 respirator group. So the N95 respirator was not notably better than just the plain surgical mask. What I would have liked to have seen is a 3-arm study where they did a negative control.
That's right.
What's the case rate in those? So the masks may or may not be helpful. Kind of like the hand washing, they probably can't hurt unless you're putting on if you do get a real respirator mask and it has one-way valves on it, just make sure you know how to purge it properly. But
I have seen, wow, these Canadians are contrary today, aren't they?
yeah,
I have seen it the other way too, Alan. I've seen a few studies where the N95s are slightly better than the surgical
Yes.
masks. But the
Yeah.
difference is slight.
yeah.
So I don't know. But it's up to you. But
And in fact, the same guy who's dissing hand washing claims that N95 masks have been proven protective.
right.
So there's a lot of debate around both of these techniques. But you can say an N95 mask costs some money. They're a few bucks a piece.
Yeah.
Surgical mask costs less money, but it's still some money. Hand washing is free, or darn close to it.
Sure.
So why not implement the thing
Yeah.
that costs nothing?
Totally agree.
Just to move back to the vaccination again, I found a Consumer Reports poll where they looked at American adults, U.S. adults. 34% said they would get the vaccine, 21% said they would not, and 43% said they would decide depending on how things go, which is not really the proper approach. It may be too late by then. So there's a lot of uncertainty, and I think part of it is because we're painting a picture that this is a mild virus. Well, it is mild compared to 1918,
Yep.
but people do die routinely from
You
flu.
can't catch the flu by being vaccinated against
No.
it.
So what's the problem?
That really has to be made more clear,
But
I think.
we have not done our job well enough.
No,
If people
you're right.
aren't taking this vaccine, we haven't
right.
taught that they should have this.
That's true.
And that's why we have to keep doing TWiV for a long time, I guess.
Now, I will admit right here, I have not yet gotten my seasonal flu shot.
Okay, why not, Alan?
Because I just haven't gotten to the. It's just I've been terribly busy.
All right. I got mine on Friday.
Yes, I was actually there's an opportunity. Our town is having a flu clinic tomorrow. Unfortunately, when I will be out of town, but I saw the sign for it and I said, "Oh, I'm going to be out of town." Well, as soon as I get back, I'll just have to go to whatever drugstore, the CVS and all those have the little.
Well, next week you might also have the pandemic flu vaccine, so you might as well get them at the same
Get
time.
them both at the same
You
time.
get one in each arm and have two sore arms.
And I will say, for my daughter, I put off until they've got both vaccines so I can take her to the doctor and get both at once.
Yeah, instead of going back, sure. You going to get yours, Dick?
Of course.
I'm going to take you to get the pandemic next week, okay?
We'll get both then. 'Cause
Yeah.
I haven't
Just
gotten
go to the
those.
lobby here. I got it.
Yeah.
That's all. All right, let's do some email. We have an email from Megan, who
Okay.
writes, I'm a few episodes behind on TWiV and just finished up with episode 44. I just wanted to comment on another career option that we have as scientists. I am trained in viral immunology, recently finishing my PhD June 2008, and have since completed a short year-long postdoc working on gut mucosal immune responses and HIV therapy. While I was completing my PhD, I realized that academic science was not suited to my interests, and I was more drawn to public health. I started contacting lab directors of state and local public health labs around the country to get an idea of the career path and, for lack of better words, what they do. I realized that this field was of great interest to me and found a public health lab in Southern California that was looking for a PhD scientist to train and eventually replace the retired lab director. I've only been here a month and a half, but I'm truly loving it and know I made the right decision. Clinical applied science is a very different beast, and I'm learning so much every day. For anyone interested, I have included a PDF explaining the education, training requirements, as well as how there will be soon a shortage of qualified lab directors in the near future. Great job security. So I'll post that PDF that Megan sent.
That's a good point.
Thanks for the great podcast. I look forward to the new one every week. That's very interesting advice. Good. That's a career path we haven't talked about.
Right.
There was
Lab
a
science.
postdoc in Howard Schumann's lab who did that, and I hung out with her in graduate school, and now I've blanked on her name.
Yeah,
Oh, well.
I know who you mean. She's actually in New York City, I think, in the New York City Health Department.
Yeah,
Right?
yeah, blonde, tall, thin girl.
Stacy? No, that wasn't Stacy.
No, not Stacy.
Somebody else.
Well,
Okay.
anyway, if we can remember her name, maybe we can get her on the show.
She's probably listening.
Yeah.
Ibrahim wrote. Now, Ibrahim is a microbiology tutor in Egypt. I just started reading the Andromeda strain. Actually, at first I thought this was a novel, though I read in the early first page that this recounts an American crisis. Going through the following pages, I am reading some unbelievable things, and I think I just can't figure things right. Is this all real? Or just nothing is real and that it's really a novel? Thanks.
Everything written by Michael Crichton should be read as fiction.
Correct. Even his nonfiction.
Well, in the first page, it said this is a recounting of an American. It's part of the fiction,
that particular
right?
novel, actually, Crichton would agree it's fiction.
It's like War of the Worlds with H.G. Wells.
The mark of a good author is that you believe that it's real.
Correct.
Right.
So then he wrote another email more calmly. Here's a link for a science article about swine flu where the statement is made. When the animals were co-infected with swine flu and seasonal flu viruses, H1N1 kept its genetic material to itself. My question is, can really different subtypes of influenza during co-infection, given these complex replication steps and segmentation of genome, keep its genome completely without any reassortment? And what are the factors that may lead to each subtype knowing its exact segments among different subtype segments? Thanks for the great show. He's referring to a paper that came out a few weeks ago which said that when you co-infect ferrets with seasonal and pandemic H1N1, you don't see any reassortment. So I think that it depends. In that case, what happened was the pandemic H1N1 just grew so much better than the seasonal. There wasn't any opportunity for co-infection. But I don't know any particular reason why co-infection would not lead to reassortment. And you could imagine that you could manipulate the amount of virus in experiments to favor reassortment. So to say that it would not occur would be wrong, I think. I think it can always occur among type A and among type B and among type C, not across types, okay? Only within subtypes or among subtypes. Okay, Jim, could you guys explain how one develops a library of 27 billion antibodies that Wayne Marasco used to identify the SARS antibody? I found many references to the library, but no discussion about how it was constructed. It sounds like a decades-long project.
Inject an antigen into a mouse.
No, what they did, Alan, is they made a library, a phage display library. So what they did is they took B cells from people, and they wanted to identify antibodies against the SARS virus. So they amplified all the antibody genes from their cells by polymerase chain reaction, and then they put these amplified genes into bacteria and grow them up as single colonies. And they're 2.7 times 10 to the 10th individual bacterial clones.
Pretty much what I said. No, they used a phage library. You're right.
So that's where the 27 billion comes from. It's not unusual in cloning to have billions of clones because bacteria grow to very high concentrations. So you can grow a flask of bacteria with billions of cells. That's why we use them.
Well, and to get to kind of the essence of this question, the reason you can get that kind of diversity in antibodies is that it's already there in your B cells.
Is it already there? Don't you get some additional diversity as the B cells are
You do
dividing?
As they mature, but
Right.
the so you've got billions and billions of B cell clones, different lineages,
Right.
and each is producing its own type of an antibody.
Right.
So they're all circulating around saying, "Hey, I make antibodies against this antigen," before your body has ever even seen it.
Right.
And that's there. That's just sitting, waiting
Sure.
for that antigen to show up. And for probably 26,999,000,000 of them, they're never going to show up. But these guys just took that entire genetic setup and cloned it into phages and used it as a display library. Now, obviously, as you point out, when you get an antigen into this system and it selects a B cell that does respond to it, then that B cell has won the lottery and it gets to reproduce, and it also produces variations
Right.
on that theme.
Right.
Which will try to get a higher affinity antibody, so you get more diversity around,
Right.
say, the particular seasonal flu that you've been vaccinated against. You'll get more diversity around that than you would around some chemical compound that's never been made.
Good explanation. So here they have 27 billion individual bacterial clones, each one making it different. Maybe not each one making it different, but there are 27 billion clones making antibodies of different sorts. The cool thing here, which Jim didn't ask about, is this phage display. It's a bacteriophage engineered, so each one displays the antibody on its surface, and then they can use that to select out the one they're looking for. And here they used it. They selected an antibody against SARS coronavirus glycoprotein, and they used people who had never been infected.
Right.
So as Alan said, these are antibodies that are there, right?
Right.
And then they got one out of these 27 billion that reacted with it, or a few. So it's very rare. Okay, so that's where the 27 billion, it doesn't take 10 years. You can do this in a few weeks. You just take the cells, B cells from people, extract RNA, you extract DNA, you amplify up the antibody genes, and then you put them into bacteria, and you grow up 27 billion bacteria. Not so bad. And then while he was looking this up, he found this link. It looks like a YouTube spin-off, and it's called DNA Tube. I've never looked at this. Have any of you guys seen this?
No.
It's sort of like a movie site, DNAtube.com, and it's a scientific video site. And here we got H5N1 transmission through pigs, swine flu, attacking influenza, flu virus animation, etc. So this is all about science of different sorts. Heart business, lysogenic cycle of viruses, cool. Development of the human fetus. So this is the big thing now in science movies. That's why we have to do a virology, a TWiV movie.
TWiV,
like
the movie.
They also have televised lectures from
Televised lectures. It's anything video science related. DNAtube.com, scientific video site.
Great.
It's a good one. So thanks, Jim. We hadn't seen that. Ed is our friend from South Africa. He's a virologist. Vincent just downloaded this. We're referring to TWiV something. I don't remember which episode. And listened. Major job. Lousy bandwidth this part of the world, South Africa. And thanks for the mention. I like the idea of vertical farming, and you might like to read this stuff for some more ideas. There's a lot of stuff going on with plant production of pharmaceuticals, less so on vaccines, but still quite a lot of promising material.
Nice.
And he sent 2 PDFs about producing pharmaceuticals in plants, which we'll post. And then he wrote again later. I had pronounced his name wrong on one of our netcasts. See, it's spelled Rybicki. I said Rybicki, but he said it's Rybitsky, by the way, which I wouldn't have guessed from the spelling, but
Nope.
What do I know? Then he says hepatitis B in bananas was a hype idea and it's dead.
Yes.
Growing plants inside is what is happening. And if you realize that there are glass houses for growing cut flowers or tomatoes, meaning it's economical, that cover tens of acres.
Yes.
And tobacco can yield a couple tons per acre.
That's right.
And vaccines could yield a gram a kilo. You realize the potential for producing a lot of vaccine in just one big greenhouse. So no need to grow outside and no controversy. Dick loves it.
He does.
And
The other thing about growing outside, I was just doing an article on antibody production for Nature Methods, and one of the sources I was talking to was talking about plant production, very similar process.
Yeah.
And I said, so what about this idea? Some people sometimes float about of farming these things just like a regular farm. And he said, well, when you have worms crawling through your production facility, the FDA doesn't like that.
You know what? There are no worms in hydroponic farming.
Right. So
Sorry
they would do
about
this
that.
all indoors.
Cool.
Absolutely, under stringent conditions.
So in that episode where we discussed this vertical vaccine farm, I think Rich Condit was with us.
Yeah.
And I asked Rich if he knew Ed, and he said, nah, never heard of him. And he said, Ed writes, and I've met Rich Condit, but he doesn't remember. I've stayed with the Moyers in Florida, too. Never been to a Gators game, but I have a shirt. So he's seen you, Rich, so don't fall off the bike,
Exactly.
Ed Rybitsky. Thanks, Ed.
Yep.
And then we'll do one more to try and keep this under an hour. This is from
That would be a miracle if you did, by the way.
BAS.
Alan and I will do everything we can to interrupt you to make sure it goes over an hour.
This is a very interesting email. Do you remember the no agenda stuff we did a couple of weeks ago?
Yeah.
Right.
All right, so this is with respect. There is a comment thread on the No Agenda forums about TWiV 44. I am trying to bridge a gap here, and I think you guys are missing a real chance for a teachable moment. My post is highlighted here, but you can see the whole thread on the same page and some of the reactions. So there's a link to this forum, so the listeners of No Agenda have their own forum for discussion. And there's a thread going on about the show we did where we criticized their criticism of adjuvants, among other things. And then he goes on, the audience of No Agenda are exactly the people who need to be reached and taught how to think critically, Being accustomed to science, it is easy for us to forget how seductive conspiracy can be as an explanation for the world's evils, and the illusion of control it gives us to feel in the know.
Right.
One important thing you were wrong about, though, the No Agenda hosts aren't peddling controversy for ratings. They both really believe what they're saying and are really trying to help people. This sincerity makes it even harder for mere facts to paddle against their current, especially when one has no expertise in the subjects at hand. I think it would be great if you could do an elementary cast for a public audience explaining virology and immunology and why all the anti-vax information is wrong. Like not just refuting the claims, but starting with why the claims superficially might seem correct or compelling, and then why they're totally wrong. Of course, I'm also aware that it's not in any way your job to reel people back from the edge of lunacy, and that one can spend a whole life shouting into the wind, and the same falsified claims will keep coming up again anyway. Thanks for doing science, even if the unwashed masses don't believe in it. BAS. So I thought about this because we received this quite a while ago, whether we should do a cast on vaccines and their danger and explain, why it's not correct that people think that they're dangerous and causing things like autism, but we haven't done it. What do you think about this, gentlemen?
Well, I think it's a good idea, but I don't know who would listen except the converted.
I think this information has already been discussed extensively. There's a lot of discussion online, and I mean we've plugged Paul Offit's book. There are a lot of people who have delved into this argument and gone at some depth and some risk to themselves in some cases to debunk this nonsense. So
Well,
yeah,
go ahead.
the response is out there, and I'm not sure that doing the same on the show is going to do any more than what Dick just suggested. I mean, we'd be preaching to the converted. All we can do is point people to the resources that are already there. And by the way, I just looked at this thread on the No Agenda forums, and I don't think that this is a teachable moment. Just the level of discussion there is, I don't even know what to say.
Exactly.
I mean, part of the problem is that crowd is they like to call people names, right, when they don't agree with them, so it gets into a shouting contest. So I'm not sure.
And I love this. If you may have to edit this for language, but I'm just reading one of the first comments that comes up in this thread. Somebody posts and says, first off, who are these douchebags, and how in the hell can someone stay awake while listening? Second, they started off after both clips with personal attacks.
Yeah, that's right.
I don't think we did we? Maybe we said these guys don't know what they're talking about.
Right. I think I may even have called them ignorant, but truth is a defense. And it interests me that somebody who starts out by calling us douchebags then turns around and accuses us of making personal attacks. So anyway,
right.
That's the level of discussion going on here. And I think we shouldn't argue with fools because somebody might have a hard time telling the difference.
Yeah.
Well, it's interesting if you go right back to the origins of the social marketing for vaccines. When the smallpox vaccine was first marketed in the United States, it was assumed that this population was different from the one in Europe, and therefore they had to do the same clinical trials all over again. And the story that I've heard, and I'm sure that this can be verified by some of our listeners, there was a banker in Philadelphia who actually sacrificed 6 of his sons to the clinical trials by vaccinating 3 of them and leaving 3 not vaccinated, then bringing them down to the hospital where there was an outbreak of smallpox, exposing all 6, and proving beyond a shadow of a doubt that the 3 that were immunized survived, and the 3 that did not were killed by the outbreak. And he said, "See, it works." Now,
old school virology.
That's a pretty harsh parent, I'd say, but I think that's not an apocryphal story. I think that was actually true. To have to go to that length to convince people that it's a good idea to vaccinate, I mean, we can't do that today, obviously.
No. The best evidence is the prevention of disease.
You don't have to put your hand in the side of,
Sure.
Well, whatever, you know what I'm talking about.
Well, I appreciate the sentiment of BAS, all right? I understand that you think we could have, but I'm not sure that they would listen to us. They're fans of the No Agenda hosts. And I really contest the idea that they're they aren't peddling controversy. I know them from other venues, and I think they are. But
No, I think that's the show's entire shtick, as far as I can tell,
Yeah.
is that they're going to say stuff that's offensive and that is fringe, and they are peddling controversy. If they weren't controversial, then I don't think they'd really have a show.
yeah. Well, I think that we don't do a specific show for that, but they have to listen to us. They have to want to, listen to a podcast, which is a little harder to absorb than usual. We have a lot of listeners, so it's not impossible. And if you stick with us for a year, you'll learn a lot. I think you may not get everything from every show, but in the long run, you're going to learn, and you'll probably realize in the end why vaccines are safe.
I think the biggest challenge to that other group, No Agenda, is the hypothesis-driven science. I mean, if you don't adhere to that as the way of testing things, then you can say anything you want and get people to believe you.
That's a very good point. And I think that we could do is teach people how hypothesis-driven science is done.
Exactly right. Exactly.
And that's not a bad idea.
Yep.
All right, let's do our weekly picks. Dick has already told us his.
I have.
That's Younger by
Judith Sulzberger. Dr. Judith Sulzberger.
And this is science fiction, and it relates to
It
aging.
is. Yep, but she had good science advisors.
Telomeres and Telomeres, Nobel Prizes.
Yep.
Good pick.
Yep.
All right. Alan.
Well, I was actually going to pick NobelPrize.org, but then I saw one of the links in the show reminded me that I had not yet picked one of my favorite science news sites. Actually, it's called ScienceLine. At scienceline.org. The shortest distance between you and science is their tagline. And I have a soft spot for this particular site. It's not exhaustive. You're not going to get all the science news you ever need. What you will get is periodic feature articles on a variety of topics related to science, and usually with a New York link. They're usually talking to a researcher who's based in New York.
All right.
And the reason for that is that they are part of the Science, Health, and Environmental Reporting program at NYU, where I used to teach. And these are journalism students.
So
these are graduate level journalism students who are practicing their craft by writing for this website.
Cool. So you know some of these writers, presumably.
I do, and I know well, certainly the folks behind it.
Yeah.
And they're they're good. I would say good kids, but they're graduate level journalism students.
Yeah. That's great.
And they do video and audio as well as written articles, and it's a wide range of topics.
Cool. Karina Storrs is the art director. Does that ring a bell, Alan?
It does.
She was a PhD student here.
Yes.
Interesting.
Reporting science to the public is the hardest job on the planet.
She's writing as well.
And Alan deserves kudos from both of us for doing such a nice job.
Well, I wouldn't call it the hardest job on the planet necessarily.
Well, to convince people of the virtue of science, and to get involved in it and to support it with their tax dollars, that's the part that's hard.
Yes.
Nice. This is a good one. I've come across this before. It's good to point out. And Karina used to listen to TWiV, so if she still is, she will.
Her ears are burning.
Pick this up. That's nice. If not, we'll let her know about it.
Yep.
And my pick is a webpage called FluView from the CDC, from the Centers for Disease Control and Prevention, which we have referred to quite a bit here. But I think it's worth emphasizing because it's the weekly surveillance report for influenza.
Right.
And it gives a summary of all the specimens that have been tested and what they have turned out to be for every week for the past years. And you can look at drug resistance, subtyping, the spread of the infection, excess mortality, really a huge site full of stuff you can go to every week and get new information. And now that the infection is ramping up again, the flu season is beginning here in the northern hemisphere, it's great to go to.
And it'll be wonderful to see the relationship between the patterns of outbreaks and the patterns of vaccination use, and to see whether it makes a difference. There will be living proof, ongoing living proof on the website. Yes. You'll have a real-time study.
And if you're one of those conspiracy theorists, you should try to develop an explanation for how somebody could create such a deep and broad base of data with such incredible internal consistency.
Did they do that for the moonshot, right?
They did.
Yeah.
Of Of course. And the
Right.
hole in the Pentagon that was done by a missile, not an airplane.
Right.
All right, that will do it. If you are a new listener to TWiV, by the way, please consider subscribing in iTunes. It's free and allows you to automatically receive each new episode as it's produced. If you don't use iTunes, you can always download your episodes at twiv.tv, and that's where we keep our show notes. If you like TWiV, tell your friends about it. Links and reviews in iTunes are particularly helpful to us. TWiV, by the way, is part of sciencepodcasters.org and promednetwork.com, which are websites where you can find high-quality science podcasts from a wide variety of fields. Gentlemen, thanks for joining me.
Pleasure, Vince.
Professor Despommier
Alan?
at Racaniello.org, medicalecology.org, and verticalfarm.com. Thank
Wow.
you very much. Alan is at dovedocs.com.
Yes.
Thanks for joining us. I hope you enjoy your meeting tomorrow.
Thank you. I
in New
hope
York
to.
City. And we shall be back again, of course, next week. Dick, will you be around?
You bet.
And Alan?
I hope to.
What is next week? What's the date? It
12th.
is the 12th. All right. I don't think we have a guest lined up, but I don't remember. We have some coming up. Do send us your questions and comments. There are 3 ways that you can get information to us. You can send us an email. You can send us an MP3 file with your email, or you can call up Skype. TWiV Podcast is our name, and leave a voicemail. And tell us everything you want us to know. We're a little behind in emails still, but that's good. It's a good thing.
Yes.
Better than having none.
Or negative emails, which
Well, that would
we
be
will
terrible.
read them, but we would get very depressed at the end of the day.
You have been listening to This Week in Virology, also known as TWiV, all about viruses. Thanks for joining us. We'll be back next week. Another TWiV is viral.